Hi friends, this is Christine K.S. Puntihan, and in this video, I will show or explain the structure of animal cell and the function of cell organelles inside it. As you all know, cells are the basic unit of your life. All the organisms are made up of one or more cells and the products of those cells. Also, cells carry out life activities like metabolic activities, requires energy growth, and they also have limited sizes. New cells arises only from the pre-existing cells by the process of cell division as stated in the cell theory. These are the differences and similarities between prokaryotic and the eukaryotic cell. So now, let's just focus to the animal cell only and talk about the components of the animal cells. There are three main components of the animal cells. First is the cell membrane, or you can say it plasma membrane. So let's just put the label here. Plasma membrane. So this is the plasma membrane of the cell, the color violet. So, every cell in the body is enclosed by a cell in plasma membrane. The cell membrane separates the material outside the cell, extracellular, from the material inside the cell, intracellular. It maintains the integrity of a cell and controls passage of materials into and out of the cell. All materials within a cell must have access to the cell membrane, or the cell's boundary, for the needed exchange. The cell membrane is a double layer of phospholipid molecules. Proteins in the cell membrane provide structural support, form channel for passage of materials, act as receptor sites, function as carrier molecules, and provide identification markers. So next is the nucleus and nucleolus. So let's put the label of nucleus here. Here. Nucleus. And the nucleus over here. Nucleolus, Okay. So this is the nucleus of the animal cell and the nucleolus of the animal cell. So the nucleus, formed by a nuclear membrane around a fluid nucleoplasm, is the control center of the cell. Threads of chromatin in the nucleus contain deoxyribonucleic acid, or DNA, the genetic material of the cell. The nucleolus is a dense region of ribonucleic acid, or RNA, in the nucleus and is the site of ribosome formation. The nucleus determines how the cell will function as well as the basic structure of that cell. So, next is the cytoplasm. Let's put the label of cytoplasm over here. So this is the cytoplasm of the cell. The color blue of the cell structure. This color blue. So the cytoplasm is the gel-like fluid inside the cell. It is the medium for chemical reaction. It provides a platform upon which other organelles can operate with the cell. All of the function of the cell, expansion, growth, and replication are carried out the cytoplasm of the cell. Within the cytoplasm, materials move by diffusion, a physical process that can work only for short distances. So now let's move on to the cytoplasmic organelles. So the cytoplasmic organelles are the little organs that are suspended in the cytoplasm of the cell. Each type of organelles has a definite structure and a specific role in the function of the cell. So let's start first, define first the raw and smooth endoplasmic reticulum of the animal cell. So this is the raw endoplasmic reticulum of the cell. Raw endoplasmic reticulum. So let's put the label over here. Raw endoplasmic reticulum. This. raw endoplasmic reticulum. And the smooth endoplasmic reticulum could be labeled or can be put in here, over here. This, this, and this. Okay, let's put the label over here. So that is the smooth endoplasmic reticulum. Okay. So the endoplasmic reticulum can either be a smooth or raw, and in general, its function is to produce proteins for the rest of the cell to function. So the raw endoplasmic reticulum, the raw endoplasmic reticulum has on it ribosome, which are small round organelles whose function is to make those proteins, while the smooth endoplasmic reticulum synthesis of carbohydrates, lipids, and steroids, hormones, detoxification of medications and poisons, and storage of calcium ions. So next is, let's define the Golgi apparatus of the animal cell. So the Golgi apparatus of this cell is this portion, the color brown. So let's put the label over here. Golgi apparatus. So the Golgi apparatus or Golgi complex is the function as a factory in which proteins received from the ER or endoplasmic reticulum are further processed and sorted for transport to their eventual destinations. Next is the Golgi vesicles of this animal cell. So the Golgi vesicles of this cell is this, the circle and the color brown. So let's put the label over here. So this is the Golgi vesicles of this animal cell. Okay. So the Golgi vesicles, its main function is the packaging and secretion of proteins. It receives proteins from endoplasmic reticulum. It packages it into membrane-bound vesicles, which are then transported to various destinations such as lysosome, plasma membrane, or secretion. And next is, let's define and label the lysosome of the animal cell, or this structure. So the lysosome of this structure is this portion. This was the lysosome. So let's put the label over here. Lysosome. Okay. So that is the lysosome of this structure. So the lysosome functions as the digestive system of the cell, serving both to degrade material taken up from outside the cell and to digest obsolete components of the cell itself. So next is the ribosome of this cell structure. So the ribosome is the color maroon, this small star shapes. Supposedly this is a circle, but I can't see any other materials to put in here. So I'd use the star shape because I have no choice. So anyways, let's put label the ribosomes over here. Okay, ribosomes. So the ribosome is an intercellular structure made of both RNA and protein, and it is the site of protein synthesis in the cell. The ribosomes reads the messenger RNA sequence and translate that genetic code into a specified string of amino acid, which grow into long chains that fold to form proteins. So that over here, ribosome. Oh, that is ribosome. So next is the vacuole. So the vacuole of this structure is this. the color yellow. So let's label it here. Put the label here. So the vacuoles are membrane-bound organelles that can be found in both animal and plants. In a way, they're specialized lysosomes, that is to say that their function is really to handle waste products. And by handle, mean take in waste products and also get rid of waste products. So this is vacuole. So next is, let's define the mitochondria of this cell structure. So the mitochondria, or the mitochondrion of this structure is this. So let's put the label over here. Oh, I guess here. Mitochondrion. Okay. The mitochondrion are membrane-bound cells, organelles that generate most of the chemical energy needed to power the cell's biochemical reactions. Chemical energy produced by mitochondria is stored in a small molecule called adenosine triphosphate, or ATP. So next is the centrioles of this cell. So the centrioles of this animal cell is this, the color green over here. Let's put the label of centrioles over here. Centrioles. So these are paired barrel-shaped organelles located in the cytoplasm, yes, of animal cell near the nuclear envelope. It play a role in organizing microtubules that serve as the cell skeletal system. They help determine the locations of nucleus and other organelles within the cells. So next is the microtubules of this animal cell structure. So let's put the label of microtubules over here. So wait, this is the microtubules of the animal cell. Next. By the way, I'd use a matched as my microtubules in this structure. So yes, microtubules. So let's put the label over here. This was the microtubules. Okay. So these have several functions. Like, for example, they provide the rigid, organized component of the cytoskeleton that give shape to many cells, and they are also major components of cilia and flagella. They participate in formations of the spindle during cell division. So those are the different organelles that can be found in the animal cell. So this. Okay. By the way, guys, when we talk about microbes, some people might think that it was a harmful organisms that might cause diseases for us without knowing that microbes really are, without knowing what microbes really are, rather. And if you see this video, you will realize how beneficial the microorganisms for us. And if you only knew that microbes can contribute to the digestion of food, produce vitamin K, promote development of immune system, and detoxify harmful chemicals. And of course, microbes are essential to making many food we enjoy. The microbes or microorganisms can also the production of antibiotics, production of dairy products, alcoholic beverage, bread making, food yeast, production of organic acids, vitamins, production of enzymes, production of steroids, dextrans, and others. But at the same time, if there are beneficial actions of microbes in our society, there are also destructive actions of microbes in our society. So, the primary harmful effects of microbes upon our existence and civilization is that they are important cause of disease in animals and croplands, and they are agents of spoilage and decomposition of our foods, textiles, and dwellings. Some destructive actions of microbes include pathogenic and food spoilage,